Welcome to the Deep Dive. Today we're diving into one of the most formidable public health challenges of our time, cancer. It's a disease that impacts so many lives, and it presents this fascinating paradox, really. We've seen incredible advancements in treatment, truly revolutionary breakthroughs. Yet despite all that progress, we're still grappling with these persistent challenges like drug resistance and, the disease coming back after it seemed like it was gone.
That's exactly right. For this deep dive, we're going to explore two critical, sort of intertwined facets of this fight. First, the groundbreaking potential of immunotherapy using the body's own defenses. And second, the stubborn reality of how cancer cells actually develop drug resistance. We'll also touch on some of the complex economics behind drug development, too.
Yeah, absolutely. Our mission today is to unpack these complexities, try to cut through some of the noise, and give you a clear, informed picture of why cancer is still so hard to cure, and, importantly, where the real hope for the future lies.
Yeah.
Okay, let's unpack this. Let's kick things off with immunotherapy. For decades, traditional treatments like chemo and radiation were the mainstays, but they often hit this wall. Cancer cells are just incredibly adaptable to develop resistance. Immunotherapy, though, it's a leading strategy now that basically trains or boosts your own immune system to fight the disease. And it's showing real advantages like better progression-free survival for patients.
Uh-huh. And what's fascinating here is how your own immune system works. It has these built-in checkpoints, think of them like natural brakes, to stop it from attacking healthy cells, which is crucial, obviously. But cancer cells, they're clever. They exploit these exact same checkpoints, things like PD-1 and CTLA-4, to basically hide from the immune system and just grow unchecked.
Right, so if the cancer cells are hitting the brakes, then these immune checkpoint inhibitors, or ICIs, they're like cutting the brake lines. They work by blocking those inhibitory signals, essentially taking the brakes off your T cells so they can wake up and go after the tumor cells. Like ipilimumab, that was the first FDA-approved one back in 2011 for advanced melanoma. It was a real game changer, targeting that CTLA-4 checkpoint.
Yeah.
Showed how powerful this could be.
Exactly. And then came the next wave, like pembrolizumab and nivolumab, targeting the PD-1 pathway. Normally, PD-1 signaling causes T cells to get exhausted, creating a more tumor-friendly environment. Blocking it really boosts anti-tumor immunity, makes T cells more cytotoxic, and can help limit metastasis. These drugs have shown, really outstanding results, especially in melanoma. And now there are others, like atezolizumab, targeting PD-L1, and even newer targets like TIGIT and LAG-3 being explored.
So we've gone from taking the brakes off to, well, almost like engineering super soldiers with CAR T-cell therapy. This is truly revolutionary stuff. Basically, they take a patient's own T cells out, genetically engineer them in a lab with these chimeric antigen receptors, or CARs, then grow billions of these modified cells and infuse them back into the patient. Now they're programmed to specifically hunt down and destroy the tumor cells.
And the evolution of CAR T has been pretty remarkable. The first generation, they had some limitations, not always super effective. But the second generation added these co-stimulatory signals like CD28 or 4-1BB, making the T cells multiply better and stick around longer. That's where we got drugs like Axicell and Ticicell for lymphomas. Now we're seeing third, fourth, even fifth generations, sometimes called TRECCAX, that carry extra payloads like cytokines to make them even more potent, especially against solid tumors, and hopefully safer too. And there's work on universal CAR T cells, UCARTs, which could be like an off-the-shelf treatment for anyone. That's a huge goal.
Right, moving beyond directly modifying cells, there are cancer vaccines. These aren't like preventative vaccines for infections. Instead, they aim to teach your immune system to recognize specific markers or antigens on tumor cells. The goal is to build this durable immunity, like training your immune system to remember the enemy long term.
Yeah, and they come in different flavors. There are cell-based ones like Sipuleucel-T, that was the first FDA-approved cancer vaccine back in 2010 for prostate cancer. It used dendritic cells. But here's where it gets really interesting now. The potential for mRNA vaccines in cancer. We all know them from COVID, but researchers were actually looking at mRNA for cancer almost 2 decades ago. There have been promising clinical results, like in melanoma and glioblastoma, and a big part of the progress has been figuring out how to deliver them effectively, often using lipid or polymeric nanoparticles, tiny delivery trucks for the mRNA.
That's amazing, isn't it? How long these ideas can be worked on before they really break through. And to make these vaccines work even better, they often use something called adjuvants. What are those exactly?
Adjuvants are basically substances added to a vaccine to help stimulate a stronger immune response. They kind of kickstart your innate immune system, which then helps the adaptive immune system, the T cells, mount a more powerful, targeted attack against the cancer antigen. Think TLR agonists, for example.
Got it. And what about cytokine therapy? You mentioned cytokines earlier.
Right, cytokines are signaling proteins crucial for immune cells to communicate and grow, like messengers. The challenge has always been that they don't last very long in the body. So scientists are engineering superkines or fusion proteins that are more stable and hang around longer to boost that immune response.
Okay, so beyond the cells and molecules, there's this whole other area opening up,
Yeah.
the gut microbiome. It sounds kind of wild, but research is showing that the trillions of bacteria living in your gut can actually influence how well immunotherapy works and even its side effects.
It's a really hot area. Certain gut microbes seem to stimulate T cell activity, for instance. Others can produce beneficial compounds like butyrate from digesting fiber, which might have anti-tumor effects.
So your diet could literally be impacting your response to cancer treatment. Incredible.
Given all this complexity and how adaptable cancer is, the real push now is towards combinatorial strategies, using multiple approaches together. You don't just throw one thing at the cancer, you hit it from different angles to overcome resistance and hopefully get better outcomes.
Makes sense. Like what kinds of combinations?
Well, combining radiation therapy with immunotherapy is one. Radiation can actually help the immune system see the tumor better by releasing antigens, though it can also have some immunosuppressive effects you need to manage. Chemotherapy plus immunotherapy is another common one. Chemo can sometimes prime the immune system too. Then there's targeting therapy plus immunotherapy. Think about drugs that block blood vessel growth in tumors, anti-angiogenesis agents like bevacizumab combined with ICIs, or BRAF MEK inhibitors with ICIs and melanoma that's been very effective.
And combining different immunotherapies.
Absolutely. Combining CAR T cells with checkpoint inhibitors like PD-1 blockers, or engineering CAR T cells to resist the suppressive tumor microenvironment, maybe by targeting TGF-beta, or even making them secrete immune-boosting molecules like IL-12 themselves. Sometimes, pre-treating with chemotherapy to temporarily deplete suppressive immune cells, that's called lymphodepletion, can make CAR T cells work better. And cancer vaccines are often combined with ICIs and adjuvants, especially for cold tumors that don't naturally attract immune cells very well. The vaccine primes the T cells, making them more responsive to the ICI.
It's interesting that you hear that immunotherapy has generally had more dramatic success in blood cancers, leukemias, lymphomas, myelomas. Why is that?
That's largely true, yeah. Hematologic cancers often express specific, easily targetable markers like CD19 or CD20 uniformly on their surface. This makes them, relatively speaking, easier targets for things like anti-cells or certain antibody therapies. Solid tumors, on the other hand, are usually much tougher nuts to crack for immunotherapy. They tend to be more heterogeneous, the markers aren't uniform. Plus, the physical structure is challenging. Higher cell density, often poor blood supply, making it hard for drugs or cells to get in. And critically, they often create this highly immunosuppressive tumor microenvironment, the TME. It's filled with cells like tumor-associated macrophages, TAMs, regulatory T cells, TREGs, and myeloid-derived suppressor cells, MDSCs, that actively shut down the anti-cancer immune response. They basically create a shield that limits CAR T infiltration and effectiveness.
Right, the TME. That leads us nicely into the other big piece, drug resistance. Why does cancer remain so hard to cure, even with these advances? It seems like the cells are just incredibly resilient. They find ways to escape therapy, even after it works initially, leading to the cancer coming back. It's like playing whack-a-mole, as you said, but with devastating consequences.
Exactly. It really boils down to evolution happening inside your body. Cancer cells are constantly changing, accumulating DNA faults. When you apply a treatment, it's like imposing natural selection. Any cells that happen to have a mutation making them resistant survive. The rest die off, those resistant survivors then multiply, and boom, you have a new tumor that the original drug doesn't work against. We call this acquired resistance. Sometimes resistance is there from the start. That's primary resistance.
And central to this resilience, I gather, are these things called cancer stem cells, or CSCs. You describe them as a small specialized group within the tumor. They divide slowly, they can self-renew, make more of themselves, and differentiate into other cancer cell types. That slow division helps them hide from chemo, right, which often targets fast-dividing cells. They're like the ninjas of the cancer world.
That's a good analogy. They're master survivors. They often live in protected areas or niches within the tumor. They can switch up their metabolism, often using glycolysis, which helps them survive in low oxygen environments. And they have this incredible plasticity. CSCs can make non-CSCs, but non-CSCs can sometimes turn back into CSCs and makes them a really slippery target.
And you mentioned quiescence, like they just go to sleep.
Exactly. They can enter this reversible G0 resting state. Basically, they just pause their cell cycle during treatment, lying low. Then, when the coast is clear, they can wake up, start dividing again, and regenerate the entire tumor. We've seen this clearly in diseases like colorectal cancer. It's a major reason for relapse.
So it's not just the cancer cells themselves, but their whole neighborhood, the tumor microenvironment, the TME, helps them resist treatment.
Absolutely critical. Tumors aren't just clumps of identical cancer cells. They're complex ecosystems with huge variety. And this TME, this niche, is constantly interacting with the cancer cells, including the CSCs. It regulates their behavior through cell signaling, the physical scaffold called the extracellular matrix, ECM, and factors like oxygen levels. It's a key player in resistance.
Can you break down some key parts of the TME that drive resistance, like hypoxia?
Sure. Hypoxia, or low oxygen, is common in tumors. It actually encourages CSCs to become quiescent, to go dormant. It also activates pathways like HI4A that promote stemness and help cells deal with stress, making them more resistant.
And what about CCAFs, cancer-associated fibroblasts?
CCAPs are often the most abundant cell type in the TME besides the cancer cells themselves. They're stromal cells, support cells, but they get corrupted by the cancer. They secrete growth factors and can even release tiny packages called exosomes containing molecules like WNTs, which can push non-stem cancer cells to become more stem-like and resistant to chemotherapy.
Wow. And TAMs, tumor-associated macrophages. They're immune cells. Shouldn't they be fighting the cancer?
You'd think so, but often they get co-opted too. They can actually secrete factors like TGF-beta and TNF-alpha that promote drug resistance and help cancer cells become more invasive through a process called EMT epithelial mesenchymal transition.
Okay, in the physical environment, the ECM.
The extracellular matrix, yeah. In some tumors, it becomes very dense and stiff. This can physically block drugs from even reaching the cancer cells. Plus, proteins within the ECM can directly signal to CSCs, activating survival pathways.
And you mentioned immune infiltration earlier, how that varies.
Right. Some tumors are immune deserts, meaning very few T cells get in. Others might be infiltrated excluded, where T cells arrive but get stuck at the edges, unable to penetrate the tumor core. Both scenarios contribute to resistance, especially to immunotherapies, so that's the environment. But digging down to the individual cancer cell, what are the specific tricks they use to resist drugs?
Oh, there are many. One classic mechanism is altered drug transport. Cells can literally pump the drug back out as soon as it gets in, using proteins called ABC transporters, like P-glycoprotein. Yeah. Or they might decrease the activation of a drug if it needs to be metabolized inside the cell to work, like with cytarabine. Or they might ramp up pathways that inactivate the drug.
And what about the drug's target? Big one. Many cancers are addicted to a specific mutated gene, an oncogene. Targeted therapies hit that gene. But resistance often comes from new mutations right at the drug binding site on that target protein. Think BCR-ABL in CML or EGFR in lung cancer. These mutations can even exist at low levels before treatment starts. Or the cell finds a workaround. It activates a completely different signaling pathway to achieve the same growth goal, bypassing the blocked target.
So they reroute the signaling.
Yeah.
Clever. What else?
They can mess with apoptosis-programmed cell death. They might mutate gatekeeper proteins like P53 or overproduce anti-apoptotic proteins like BCL2, essentially refusing the self-destruct signal that chemo or radiation tries to trigger.
Yeah.
They can also enhance their DNA repair mechanisms. Many drugs work by damaging DNA. Resistant cells just get better at fixing that damage, sometimes using enzymes like ALDH1, which also reduces damaging reactive oxygen species.
And metabolism. You mentioned they can switch fuel types.
Yes, metabolic adaptation is huge. Chemotherapy puts stress on the cell's energy production, so they might switch from the typical cancer metabolism, the Warburg effect, towards using mitochondria more efficiently oxidative phosphorylation, or OXPHOS. Proteins like SIRT1 are often involved in this switch.
And microRNAs. These tiny molecules seem to be involved in everything.
They really are. miRNAs are small RNA molecules that don't code for proteins but regulate the expression of other genes. They play a massive role in modulating drug sensitivity and resistance, especially in CSCs, by tweaking signaling pathways. Targeting specific miRNAs is actually a really promising therapeutic strategy being explored now.
It's just layer upon layer of complexity. Now, going back to immunotherapy for a moment, while it's revolutionary, it's not without its own challenges, particularly side effects, right? These IREs.
That's correct. Immune-related adverse events, or IREs, are a significant concern with checkpoint inhibitors especially. Because you're taking the brakes off the immune system, it can sometimes start attacking healthy tissues too. This can lead to inflammation in almost any organ. Skin rashes, colitis, hepatitis, pneumonitis, endocrine problems like thyroid issues or diabetes, even autoimmune conditions.
And sometimes more severe things.
Yes, rarer but more severe reactions like cytokine release syndrome, CRS, or neurotoxicity, ICANS, can occur, particularly with CAR T therapy, but sometimes with ICIs too. They require prompt recognition and management.
So managing these IREs is crucial.
Absolutely. It often needs a multidisciplinary team, and treatment might involve holding the immunotherapy and using immunosuppressive drugs like steroids. Finding that balance between activating the immune system against cancer and preventing it from harming the patient is key. And as I mentioned, developing next-gen therapies with better safety profiles is a major research focus.
Okay, so zooming out now to the systemic picture. We've talked about the science, but what about the money? Cancer drugs are big business.
Huge business. Cancer biopharmaceuticals represent a massive global market generating enormous revenues. This definitely fuels R&D, which is good, but it inevitably raises serious questions about affordability, access, and ultimately the value these expensive drugs provide to patients and healthcare systems.
Right.
And the U.S. system is particularly relevant here. Its legislative framework allows for what some analysts call infinite pricing power, which has knock-on effects globally. The numbers are pretty staggering. One analysis looking at drugs launched between 1997 and 2015 estimated the mean R&D cost per approved cancer medicine at around $4.4 billion. Now, that hides a huge range. Some were much less, some much more, over $15 billion for specific drugs.
Wow, $4.4 billion average.
But here's the kicker. The estimated return on investment, the ROI, for those medicines ranged from 435% to 551%. And for certain blockbuster drugs, the ROI was astronomical. We're talking nearly 2,800% for erlotinib, over 3,400% for trastuzumab, 2,500% for rituximab, 3,200% for bevacizumab.
Those numbers are hard to comprehend. And you mentioned precision medicine earlier, the idea of targeting smaller patient groups. Does that help with costs?
You'd think it might lower overall R&D costs, but there's really no evidence that translates into lower prices for you, the patient, or the payer. It seems the pricing is driven more by what the market will bear rather than actual development costs.
So this leads to concerns about the influence of the private sector.
Yes. What some researchers have termed the private capture of the public in cancer drug development, the pharmaceutical industry increasingly dominates the entire process, from basic research funding priorities right through to running the pivotal clinical trials that determine approval. They effectively set the research agenda.
And that raises questions about the trials themselves.
It does. Concerns exist about the increasing use of surrogate endpoints, things like tumor shrinkage on a scan, to get drugs approved faster, even if those endpoints haven't been definitively proven to translate into patients actually living longer or feeling better. There have also been accusations in some cases of, well, manipulation of data, overstating how well a drug works, and downplaying its side effects. The case of everolimus was cited as an example where toxicity might have been underreported initially.
What about research after a drug is approved? Does that help clarify things?
That's another issue. Post-marketing research, sometimes called real-world evidence, is meant to see how drugs perform in routine clinical practice. But often, the quality of these studies isn't high enough to really provide robust guidance for doctors and patients. Meanwhile, public institutions, universities, cancer centers often end up acting almost like subcontractors to the industry. Public research funding increasingly aligns with private sector R&D priorities, potentially reducing scrutiny on the real value of the research, or overlooking potential conflicts of interest, like direct payments from drug companies to the oncologists who write treatment guidelines.
So where do the regulators fit in? The FDA, the EMA in Europe? Aren't they supposed to be the gatekeepers ensuring drugs are safe and effective?
That's their mandate, yes, to protect the public good, but studies suggest they've struggled to consistently ensure that newly approved cancer drugs offer truly meaningful clinical benefits compared to existing options, or just supportive care. This raises an important question. For example, one study found that nearly half of the cancer drug trials approved by the EMA between 2014 and 2016 had a high risk of bias in their methodology. Another analysis of FDA approvals for solid tumors between 2017 and 2021 found only about a third demonstrated a significant clinical benefit based on validated criteria.
That's quite concerning. So even with approvals, the actual benefit might be marginal for many patients.
In some cases, yes. And despite growing awareness and efforts by groups like the World Health Organization with its essential medicine list, or initiatives like Choosing Wisely and Common Sense Oncology trying to promote higher value care,
The hike continues.
the hype and hyperbole around many new cancer drugs seem to have actually worsened, if anything. The system shows very little sign of fundamentally changing course, largely due to the immense financial interests involved. Money talks, unfortunately.
Which brings us back to that fundamental question. Why haven't we cured cancer yet?
Well, the most important thing to understand is that there is no single cure for cancer waiting to be found. Cancer isn't one disease. Each cancer type is different, and even within one type, like breast cancer, every patient's tumor is biologically unique. It behaves differently. So a treatment that works wonders for one person might do nothing for another. There's no universal silver bullet.
And there are practical challenges too, right? Like early detection.
Absolutely. For many cancers, we still lack effective screening tests for early detection in the general population. Uterine cancer is a good example. There's no standard screening, and the symptoms can be vague, meaning it's often diagnosed at later stages when it's harder to treat. This highlights why personalized treatment plans are so crucial.
We also need to talk about diversity in research.
Yes, historically, clinical trials have predominantly enrolled white patients, often excluding older adults or those with other health conditions. This severely limits our understanding of how treatments work or don't work in the diverse populations who actually get cancer in the real world. And simple barriers to access, like needing transportation to a trial site or being unable to afford taking time off work, prevent many people from participating in research or even getting state-of-the-art care. These inequities slow down progress for everyone.
And fundamentally, it comes back to understanding the biology.
Exactly. Without a deep genetic understanding of why a particular drug works in which specific tumor types or patient subgroups, it's incredibly hard to predict who will benefit. This leads to many drugs failing in large, expensive late-stage trials because they only work for a fraction of the patients enrolled. We need better biomarkers and a deeper biological understanding up front.
We have certainly covered a lot of ground today. From the really cutting-edge science of immunotherapy harnessing the body's own power, to the incredibly complex and frankly stubborn ways cancer develops resistance, using mechanisms like cancer stem cells and manipulating its microenvironment. And finally, grappling with those intricate and often troubling economic forces that shape how